113 Questions About Evolution with John Perry. Evolutionary question number 25. Endogenous retroviruses. How do we know they really came from viruses? A little while back, I published a new animation over on the Stated Clearly channel about endogenous retroviruses. And these are retroviruses, so retroviruses are a special type of virus that inserts its DNA into its host's DNA. They actually start out with an RNA genome, convert that into DNA, insert it into the cell. And when that cell reproduces, the DNA of the virus will be reproduced as well. And so now both the daughter cells have a copy, identical copy of the virus genome. They can make viruses, pump out viruses, and, the virus reproduces. Well, if they get into a sperm cell or an egg cell, the child that develops from that sperm or egg cell, if a child does develop from them, will have the virus genome in every single cell of its body. And then it will pass the virus genome on to its kids when it has kids. Our genome, the human genome, is filled with virus genomes. We call them endogenous retroviruses because they are in us from the start. So I did a video about this on the Stated Clearly channel. And I've gotten a lot of questions about some of the details that were not covered or were sort of quickly gone over in that animation. It was a very quick introduction to endogenous retroviruses and how they can be used to make evolutionary trees, because you can actually see that, humans and chimps, for example, share a bunch of endogenous retroviruses, and those were inserted before the chimp-human split. So here are the questions. Well, some of the questions. I've gotten a lot of them. I'll probably be doing a couple of videos on this. Today, I'm combining the questions that I got from two different people. One of them is YouTube user MM. How do we recognize that a portion of our DNA is an old contamination of a retrovirus? And then Rodrigo asks, could it be that the things we call endogenous retroviruses are actually just parts of our DNA that God put there at the moment of creation? Now, once we understand how it is that ERVs are recognized, ERVs is endogenous retroviruses, but it's easier to say. So once you know how ERVs are recognized in the genome and you think about it real carefully, you'll realize that they can't tell us whether or not God exists, right? But they actually can tell us whether or not they were put there at the moment of creation or not. So yeah, we will examine those questions here. The information that I'll be giving you, a lot of it comes from this book. It's a really great overview of just, the past 50 years of the study of these things. And it's by John Coffin, Stephen Hughes, and Harold Farmas. John Coffin actually was the main researcher who advised me on that video. So it was really cool being able to work with him. It turns out that if you read his book, he doesn't really divide things like I do here, but there are 11 different ways, independent ways that we can tell that ERVs really came from viruses. So the first one, we're going to go through all of these one by one. The first one, ERV protein genes match those of retroviruses. So here's a little sketch of a retrovirus. There's the outline of a retrovirus. Retroviruses have RNA for their genome. I've got this little coiled up RNA inside the actual virus particle. It's going to go try to infect somebody here. And here I have the same genome kind of laid out in schematic form. And I've got it divided into 3 big chunks. People will divide the genome into several different ways, but I've divided it in, 4 chunks, a little tail on the end there. A retrovirus has a segment that codes for GAG genes, so group antigen genes, and then a protease. The group antigen genes, those are the proteins that form a little capsule around the RNA genome. And some of them bind actually to the RNA. Some of them form a shell around it, an extra coating around it. And then there's a protease, which protease is a protein that breaks apart other proteins. Then there's another segment of the genome that codes for the polymerizing proteins. There's a really special protein that retroviruses code for called reverse transcriptase. That's what, when these viruses reproduce, they actually make a copy of their RNA genome into DNA, and then they insert that DNA into the cell. They actually slice a part of your DNA, pull it apart, and they insert their genome, a DNA copy of their originally RNA genome, into your DNA. That integration happens with this enzyme called integrase. So you've got reverse transcriptase and integrase. They're very important. Then, last but not least, we have the envelope genes. You've all heard of spike proteins by now because of COVID. These are the things that are on the outside of something like the coronavirus, and also HIV has them, the flu has them. They are the things that stick to your cells and allow the virus to break into those cells. They stick to the outside of your cells and help it, get inside. There's a bunch of different envelope genes that are coded for in a retrovirus genome. And all of those are also found in ERVs. So these chunks of DNA in our genome, in the human genome, that look just like virus genomes, we call them endogenous retroviruses. They have the GAG pro genes, the POL genes, the ENV genes. They've got all of them, and they're in, similar orders. Now, if you start reading papers on endogenous retroviruses, you will find claims that, the human genome, 5% of it is endogenous retroviruses. Some people will say 8% of it is endogenous retroviruses. And the reason that you see different numbers in different places, well, some of it's because we've discovered more of them as time goes on. We keep discovering new endogenous retroviruses. But also another reason is that some people might be only counting an ERV if it's a complete ERV, the whole thing is there. Some people might count it if it's only partially there, because what happens over time is these things mutate. So I've kind of darkened out some spots here to show the mutations. And this one is, it's pretty mutated in my example here, but the whole thing's intact. Sometimes you'll get a mutation that knocks out a big chunk of one. And then you'll get some mutations that are actually pretty common where the white part here, the part that I've drawn in white, the tail on the right will connect to the tail on the left. And actually the whole thing will get sliced out. So that happens. But some people want to count any chunk of an ERV as an ERV. Others only count it if the whole thing's intact. There's about... Again, this number keeps growing, but if you look at the databases right now, you'll find that there are about 9,000 complete ERVs that are over 90% still fully intact. So there's absolutely no question that these things are viruses, the remnants of viruses in the human genome. And they're just there, like all of us have them. So it's pretty cool. ERVs are bookended by long terminal repeats, just like retrovirus insertions. So here I've got the long terminal repeats. Up top is the ERV that's in the human genome, and then down below is a retrovirus genome, the RNA genome, and it's got one long terminal repeat on the right side here. It's got a tail there. And what happens is this enzyme called reverse transcriptase, when it's making a DNA copy of the chain of RNA, it actually copies that tail onto both sides. And so the processed DNA genome of the virus has two tails, two long terminal repeats that are identical to each other because that enzyme uses one to make the template for the other. So they're identical. And then that gets inserted into the victim's genome. That is an obvious sign that there is a virus insertion that's happened, that you've got these repeating stretches of DNA on either end surrounding the virus protein encoding genes. And those can be several hundred to over 1,000 nucleotides long, the long terminal repeats. It just kind of depends from different virus species to virus species how long that's going to be. Number 3, retroviral integrase fingerprints are found before and after each ERV. So let me explain what that means. Here I've colored in blue these little ERV fingerprints or retrovirus fingerprints. And down below I have the retrovirus genome here. So this is the DNA version, the processed version of the retrovirus genome. And below I have host DNA before insertion. So this is they haven't yet been infected by the virus. Well, what's going to happen is that little enzyme integrase that I've colored in red here in my model, it's going to make a Z-shaped slice in your DNA. And then it's going to separate your DNA. So you've got this little jagged end on both sides. And if we were to zoom in on that, it would look like this. So that's what the cut looks like. And then the virus DNA will be inserted between those two cuts. And if we were to zoom in just on those two cuts and bring them in close to each other, we see, so here, the white here, those are the long terminal repeats. This is the host's original DNA with the weird cut going on there. And whenever you have a cut in your DNA like this, a break in your DNA, there's an enzyme that's going to run over and try and fix it. And it's going to use whatever's here on the bottom. It's going to put the opposite on the top. So in my color scheme here, I've shown that green pairs with yellow and blue pairs with red. And so this enzyme will actually find the missing pair and rebuild it. And what happens is you get little identical copies of the pre-insertion site on either end of the retrovirus insertion. And let me just show you that these are identical here. So our sequence on the top is yellow, then green, then red, then blue, then yellow, then red. So the way that this process works, it ends up making two identical copies on the ends of the retrovirus insertion. So down here below, we see what a retrovirus insertion looks like. Up above, we see what ERVs look like in our genomes, these old things that scientists tell us are the remnants of ancient viral infections. Well, obviously they are. They're identical. Not only do they have the same protein coding genes on the inside of the genome, they have long terminal repeats, and they have these little scars on the ends there. So that question that was asked, could it be that the things we call endogenous retroviruses are actually just parts of our DNA that God put there at the moment of creation? Well, again, I'm not going to tell you whether or not you should believe that God exists. I've heard people say that God is controlling all things. He's holding the universe together or whatever. So I suppose that every time a virus inserts its genome into a host, that was God doing that. So I'm not going to tell you whether or not this tells us anything about the existence of God, but it does tell us that these insertions did not happen at the moment of creation. These things were put there by an integrase molecule. And integrase molecules are things that retroviruses have. So endogenous retroviruses definitely were not there since the beginning. For any endogenous retrovirus that you find in the human genome, if you go back in time far enough, maybe you have to go back to like before we were even primates, but you would find a time where that specific endogenous retrovirus did not exist in the genome because it had not yet been inserted there. These things were inserted after evolution was already up and running and our ancestors were evolving. So, number 4, some ERVs can still produce full-fledged viruses when cells are stressed. So this is an obvious indication that ERVs really did come from viruses. This is a study on mice. In this paper, they witness a retrovirus get turned back on and start producing viral particles. In pigs as well, they've got some that will produce viral particles. In live pigs, the cells in live pigs are actually, they suppress viruses. The viruses aren't usually made. But if you do a cell culture of pigs, or if you have like an organ of a pig that you're keeping alive in a petri dish or something, it'll start to express viruses because it gets stressed. It can't stop those viruses from forming, so they do. Number 5, populations vary in number of ERVs, meaning they are continually inserted and deleted. So what I mean there is that if you look at any one species of animal, and then you look at different populations of that species, you will find that they have different numbers of ERVs. I mean, most of them are identical. Most of them are shared, but sometimes you'll find like 1 or 2 ERVs in one population that are unique to that population and only found there. This is the case in chickens. So here is a study where people took a bunch of different chicken breeds and analyzed their DNA, and they found that some of them had more ERVs than others. Well, they found one that had an extra ERV. And what that means is that either an ERV was inserted in that population since humans started breeding chickens, or it means that that one ERV was somehow deleted in all other populations of chickens. And so in this case, because one population of chickens had it and all the other ones didn't, obviously the most parsimonious conclusion is that this is a new insertion in that one population. This is another thing showing us that endogenous retroviruses really do come from viruses, because how would it have gotten an endogenous retrovirus if it didn't come from a retrovirus? Koalas are just loaded with ERVs, and any individual might have way more ERVs than another one. Any family line, I should say. Because they have the viruses that are giving them these new ERVs, they're highly active. They actually make koalas sick and they get each other sick. And these things just are really good at inserting themselves in the germ line. So that they're good at getting into sperm or egg or, newly fertilized fetuses, which makes it so that they end up getting in the germ line and they're passed from parent to child. And this is happening in koalas right now. In humans, it is not happening right now, so far as we can tell, but it was happening until fairly recently, probably somewhere around 6,000 to 10,000 years ago. We still had viruses that were active and actively inserting themselves into the reproductive lines of humans. And we know this because there are certain populations that have ERVs that other populations do not. When we look for the insertion sequence, that little sequence that's copied on either side of an ERV or a retrovirus, when a retrovirus is inserted, we actually find the uninterrupted insertion spot in most humans, and in certain groups, we find the endogenous retrovirus there. So that tells us that these things have been inserting themselves until very recently. Number 6, new ERV insertions have been directly observed in the lab. So this is an experiment that they did on mice, where they would just try and get mice sick with viruses and see if they would end up inserting themselves into the germ line and actually be passable from parent to child, through reproduction. And the answer is yes. In this one, the sure way to make it happen was to take a freshly fertilized egg cell, so sperm has just fertilized it, and then expose it to a virus. And the virus will end up in all of its cells, including the sperm or egg cells of that developing fetus, of that mouse when it's finally born. And then it can pass on to its own children, that virus. So we know that it can happen. We know that retroviruses make it into the genome, and we know how they do it because we've played with it in the lab. We've seen this in chickens as well. And then another study on mice. This one was not lab induced. So the scientist wasn't actually poking around and messing with things to make sure that it would happen. What we did is we just were breeding mice, inbred mice for long periods of time, because labs like to do experiments on mice that are inbred because their genomes are all identical. So if you do something on one mouse, it's sort of like doing it on another mouse. But periodically they will sequence the genomes of their lab mice and see if any big mutations have happened. Because if big mutations have happened, they need to kind of, hone those in so that pretty much every mouse is identical. Well, they found that in these labs that were doing very, very careful breeding of their mice, to not ever let any contaminant from a wild mouse get in there. They found that within their own lab, over the course of 140 generations, they realized that some viruses had inserted themselves in some of their mice and not other ones, and they were inserted into the germline. They had become endogenous retroviruses in certain family lines of their originally inbred population. Now, one of the doubts that people have when they first learn about ERVs is how could anything survive getting a virus permanently inserted into its genome? That's absurd. Well, we know that insertions are quieted by the enzymes of the cell. So cells can actually shut these viruses down early on, and that's been observed. That cells will try and silence a virus, make it so it can't do its naughty virus stuff, even though it's in the genome there. And yeah, there's a bunch of studies on that. And what that is, these are adaptations that we must have evolved because this is a common thing. We have these viruses that pop up in our genomes and we have to deal with them. So these adaptations exist in us already. Number 8, ERV deletions have been observed in the lab. Another question that people often have is if viruses can sometimes insert themselves into a genome, even if it's a fairly rare event for that to happen, over time, they should accumulate until your entire genome is just nothing but virus genes. And no, there are ways that these things can be deleted. One way is for just a huge deletion in your genome to happen, which deletions happen randomly from time to time. And that can cut out a huge chunk of DNA, including the virus. And that's one way that things can get cleaned up afterwards. But if you remember, I said that there are these long terminal repeats on either end of the insertion. And those, when a cell is reproducing, those can get matched up with each other and the virus can get ripped out and deleted that way. And that's much more of a precision cut. It does leave one of the long terminal repeats behind, though. So there's a lot of single long terminal repeats that are just like, you'll find them all over the genome. And here's an actual study where that was observed happening. In theory, we knew that was probably what was happening, and then we've actually seen it now in the lab. So that's pretty cool. Number 9, a mouse ERV reverted into a full-fledged virus with just one mutation. So again, most of these ERVs, especially the old ones, they're mutated to the point that they don't work. But in this experiment, there was a molecule they were giving mice, and it turns out that a lot of the mice that got this molecule would end up expressing viruses, endogenous retroviruses, as if they were normal, as if it were a fresh infection. And I'm like, why is this molecule causing this reaction? Well, this molecule happens to be highly mutagenic. If you give it to enough mice, some of them will happen to have a mutation in one of their retroviruses that turns it back on. There's a lot of endogenous retroviruses in a mouse genome. And if you go poking around, you're going to accidentally turn one back on. You're going to accidentally cause a mutation that reverses a mutation in the past that disabled the virus. These are definitely viruses, all right? Now, those of you who are listening carefully, you might think that I just contradicted myself because I said that most animals have the ability to silence endogenous retroviruses. They can silence these virus genomes, and I just told you here that a single mutation turned an old mutated endogenous retrovirus back into a full-fledged retrovirus. Shouldn't the body's cells have just silenced it? I mean, how did this happen? Well, the silencing mechanisms are only good at silencing the endogenous retrovirus in certain parts of the genome, and it only appears to work in developing embryos. So if you're an adult and you get a retrovirus, you usually can't silence it. It's a special thing that babies can do. Number 10, a virus genome was deduced from human ERV sequences and built in the lab, and it actually worked as a real virus. So here's the study on that. What they did is they looked at a bunch of endogenous retroviruses that were closely related in humans, and they got what is called a consensus genome for that group of viruses. We look at them and we say, oh, look, there's a mutation that this one has and none of the other ones have. Therefore, if we edit it back to be the same as the one that all the other ones have, we've healed this endogenous retrovirus. It's now more similar to the actual virus that originally infected our human ancestors. And they did this, they had several different endogenous retroviruses that were closely related to look at and to do this. And they were able to correct every single one of the mutations that had happened since insertion, or at least enough of them that the thing worked. They put it inside human cells in petri dishes, so they didn't infect a human with it. Thank goodness. It can infect human cells. It really infects them, and it really spreads from cell to cell. So that's crazy. We brought a virus back to life from the dead, from extinction. Yay! Just what we need. More viruses. And then last but not least, new ERV proteins and new ERV regulators can give immediate benefits to hosts. And this answers the question that a lot of people have, why wouldn't natural selection just delete these things? Why is it that these things end up in our genomes? Well, it's because oftentimes they can be beneficial. And there are some really cool studies on this. Sometimes it's actually good to get a virus permanently stuck in your genome. Sometimes. The details on this are pretty cool, and this deserves its own video. So I'm not going to go over these in depth right now, but you can just take a look at these papers if you want to read them on your own, if you're extra ambitious, or you can wait until next week when I talk about them myself. And it's really cool. It's really cool what these things can do for a host if the host can stop them from actually producing full-on virus particles, full-on infection, that's going to be detrimental to them. So yeah, endogenous retroviruses can be beneficial. Let me just wrap this up here because I've been going for a while. How do we recognize that a portion of our DNA is an old contamination of a retrovirus? Well, the 11 things I just showed you, that's how. And then could it be that the things we call endogenous retroviruses are actually just parts of our DNA that God put there at the moment of creation? The answer to that is no. Whether or not God exists, again, that's up to you if you want to believe whatever. But these retrovirus insertions, these endogenous retroviruses, certainly were not there from the beginning. They have been inserting themselves into our ancestors as we started our evolutionary journey. This is a continual thing. We know this because of the signatures of integrase, which is the enzyme that integrates them into the genome of a host. And we know this because different populations of the same species will have a few endogenous retroviruses that other populations don't have. That tells us that these things are continually being inserted into our genomes. So, next question.